Starting.
Buenas noches. Lo siento no poder hablar con ustedes en español. Estoy aprendiendo español, pero aprendo lento y mal. So I will switch to English, which I may be accused of speaking poorly, but I've never been accused of speaking slow or learning slow. So I'm particularly happy to be speaking in Buenos Aires. I wish I were in Buenos Aires. My family and I had a wonderful visit to your country in January of 2022. We went to a wedding in Patagonia, and then we spent 4 days in your beautiful capital city, which was the happiest place I've seen, but that was probably because it was 3 weeks after La Copa del Mundo. So Buenos Aires seemed very happy for good reason. Felicitación en su fútbol. What I want to talk about today, I'm using a title that I stole from a famous movie that was directed by Sergio Leone and starred Clint Eastwood, who lives about 100 kilometers south from me. It's a movie from the 1960s. In English, it was called The Good, the Bad, and the Ugly. In Espanol, it would be, I think, el bueno, lo feo y no, well, anyway, the good, the bad, and the ugly. Lo bueno, lo malo, y lo feo. See? Because in bioscience, the ethical, legal, and social issues in bioscience, some are good, some are bad, some are ugly, although rather than ugly, I'm going to change that word for this talk to impossible. So what I want to do is talk about three things. I want to talk about the benefits of advances in the biosciences, both in general terms, but then with very specific technologies. Then I want to talk about some of the risks and dangers, some of the bad things that could come out of it. Again, both with in general terms and then with some specific examples. And finally, I want to talk about at least two examples of things that are talked about a lot, but that I think are fundamentally impossible, at least for the lifetime of anybody listening to me during that period. So let me start with the good. I think the benefits of the amazing advances in biosciences, and they have been amazing. Basically, we know compared to 30 years ago, infinitely more. I think that 30 years from now, they will look back at 2024 and say they knew nothing, those fools. They thought they understood things. But the learning curve has been almost vertical. And there have been at least two, in a general sense, two huge potential benefits from that. The first and the most important is the potential and the reality of relief from suffering both human and non-human. Now, some might argue that relieving suffering is not always a good thing. And I think there are examples of situations where suffering has led to good. Suffering has sometimes made people do better things. Maybe it's made countries and cultures better. Sometimes that happens, but not always. And one thing we know that suffering always causes is suffering. It's always a bad thing. The fact that sometimes it may lead to good things, I think, is not a good argument to try to stop trying to prevent suffering. But if you are worried about the elimination of suffering from the world and how that would negatively affect human character, I can reassure you that nothing in bioscience is ever going to eliminate all suffering. We will still have suffering. If we cure cancer, if we cure Alzheimer's disease, if we cure all sorts of terrible things, people will still find a way to suffer the way, say, the loser of the World Cup suffered, or worse. So I think that although we can never completely eliminate it, it is not only a good thing, but it is a moral obligation as far as we can to try to relieve suffering within limits. I'm a parent. Happily for me, my children are now 36 and 32 years old. But when they were little, they probably thought I inflicted some suffering on them. Do your homework. No, you can't watch TV. No, you can't do this. Um, Maybe they did suffer. So occasionally suffering is a good thing, but in general, relieving suffering is a better thing. And bioscience is allowing us to do that. The other benefit is a little more abstract, but to me at least, quite real. To me, and I think to a lot of people, I think for some of us at least, knowledge is good in itself. Understanding things is satisfying. Sometimes it leads us to be able to do good things, but even if it doesn't lead us to do good things, just understanding, knowing, having the facts can be really helpful, whether it's the role of genetics in Alzheimer's disease, whether it's the nature of dark matter and dark energy in the universe, whether it's understanding recent modifications in the application of the handball rule. Knowing is good. Understanding is good. And we have understood so much more about biology than other things. It doesn't mean we have fewer questions. A friend of mine thought a beautiful analogy the other day. He said, the expansion of science is like the expansion of a balloon. The more we know, the bigger the balloon gets. The balloon gets more and more full. But as the balloon gets more full, if you think about the surface of the balloon as the questions, the bigger the balloon, the more questions we have. So as we learn more about science, we also have more questions stemming from science. I don't think we'll ever answer all of them, but filling that balloon, knowing more, is in general a good thing. One does have to weigh against that the cost of pursuing knowledge. If it's going to cost $100 billion to try to do a new linear accelerator to discover something more about subatomic physics, maybe that's not worth it. If it's going to risk total annihilation of the universe, well, to do an experiment, maybe that's not worth it. So we do have some limits on when adding to knowledge is good. We need to weigh the costs and the benefits, but there is a benefit there. Plus, there is always the unforeseen benefits of where basic knowledge leads to applications that are beneficial that we did not expect. I think my favorite example of this is the wonderful genetic tool CRISPR, clustered, regularly interspaced, short palindromic repeats. Jennifer Doudna and Emmanuel Charpentier, Jennifer from University of California at Berkeley, about 50 kilometers north of me, and Emmanuel from Switzerland, won the Nobel Prize in 2020 for their discovery published in 2012 of how to turn CRISPR into a tool that humans could use to learn things and change things in biology. But they didn't invent CRISPR. Jennifer will tell you she didn't invent CRISPR. CRISPR was invented by some bacteria 2 billion years ago. Now, she doesn't think the bacteria should have gotten the Nobel Prize, but the bacteria invented it. And before Jennifer, 12 years before their publication, a Spanish microbiologist named Francisco Mojica, who works on the Mediterranean coast in Valencia of Spain, was studying some strange bacteria that he found in the Mediterranean. And he saw some very odd things in that bacteria. And it led him to discover CRISPR. CRISPR as the bacteria use it, which is as a defense against infection from viruses. That was an interesting discovery. Nobody thought it was particularly important. This is this strange little thing bacteria use to protect themselves. It took 12 years and other scientists to say, wait, we can use that discovery and turn it into a tool that humans can use to discover more and to treat disease and to change things in ways that are beneficial. So sometimes even the most apparently pure basic science with no applications that you can imagine turns into something that can be very useful. So those, I think, are the two great benefits of our, to confirm that my speaking language is English. My speaking language is English. Those are the two great benefits that knowledge of biosciences is giving us. I want to get into some more specific ones. And I do think that this is an amazing moment. We are almost at the end of 2024. We've almost finished the first 25 years of this century. And as somebody who remembers very vividly Y2K, it's amazing to me that this century is almost a quarter over. But the other amazing thing is if I look back at the year 2000 in biology, we had a great deal of confidence. We were just on the edge of sequencing the whole human genome, and that was going to allow us to understand and cure every disease with a genetic component. We had just created, discovered, trained through a scientist in Wisconsin named Jamie Thompson, human embryonic stem cells. And that was going to allow us to cure paralysis from spinal cord breaks, to fix Alzheimer's disease, to build new hearts and lungs and livers and kidneys from people. And in the neuroscience world, we just started using what's called functional magnetic resonance imaging, which for the first time gives us an opportunity to see on a moment-by-moment basis what's going on inside the brain of a healthy person. Before, if you wanted to know what was happening inside a brain, you had to take the brain out of the skull, cut it up into little pieces, look at it under a microscope. You can't really legally do that with a living human research subject. But with fMRI, you can put somebody into an MRI scanner, have them experience something, have them listen to a little bit of a song, and see what parts of their brain react to that song. And so that led us to believe in 2000 that pretty soon we would understand the brain. Well, I am planning to write an article, the title of which will be "It's More Complicated Than We Thought," because none of those things have come to pass yet. The people who were so optimistic 25 years ago, they weren't wrong. They were just too optimistic. I think they were early, but not wrong. Biology is just a lot more complicated than we thought it was. And one of the advantages of all three of those tools is they've helped us understand how complicated it is. My own hope is that it's not infinitely complicated. At some point, we get down to levels of truth that we can actually act on in useful ways. And we've done that to some extent in genetics, in stem cell research, in neuroscience, but not nearly as much as we had hoped. On the other hand, what we have done in the last 25 years is open up a set of possibilities that 25 years ago looked like science fiction. Today, they are still not yet here, but they are plausible. And I'm going to give you some examples. One is in organs. So heart, kidney, liver. Consider diseased organs. People often die from diseased organs. Well, for 50 years now, 60 years now, we've been able to transplant organs, but still many people die every year who couldn't get an organ transplant because there weren't organs available to transplant, or who had damage from the immune suppression therapy that was necessary for the organ transplant. Organ transplants have helped some people, but it's a drop in the bucket of all the people who could benefit from getting new hearts, new kidneys, new livers, new lungs. We are now coming up with new approaches. We are discovering new ways to repair or protect organs. Sometimes it involves gene therapy. If a kidney is diseased because there's a problem with some of the sequence of some of its genes, we can, in theory at least, go in and change that genetic sequence and fix that kidney. Sometimes it's just small molecule drugs, traditional drugs. But now that we understand more about what's going wrong because of the new tools, we can say, so if we can find a drug that fits this particular lock, if we can make a drug that is like the key to this particular lock, we can fix this organ. We're also finding new ways to preserve organs, to keep them alive longer, to be useful for transplant. So right now in the United States, about half the time when somebody dies and has donated their organs for transplant, about half the time the organs can't be used because by the time you get the organ from the person who's donating it, from the dead body that donated it, to the person who needs it, the organ has been damaged. We're now figuring out how to keep those organs alive outside the body to keep them fresh and available for transplant longer than we did. There's also a procedure called normothermic regional perfusion, which after somebody is dead, after someone has been declared dead as a result of their brain being dead, it keeps the rest of their body, including all of the organs in the rest of their body, alive inside their body. And then you can bring the person who needs the transplant to the person, the dead person with the living organs, who has the organs to give. That's another advance. We're also trying to figure out ways to replace organs with machines. There are some implantable sort of pancreases to deal with diabetes. So you implant a device that measures how much insulin is in your blood and releases insulin into your bloodstream when it's needed, instead of having to do a finger prick, see what your insulin levels are, and then inject yourself with insulin. A machine inside your body can now do that, at least to some extent. It's not yet fully operational, but it's getting close. Or we can replace organs, human organs, with non-human organs. There have been several examples recently of people who have received pig organs. Now, two people have received pig organ heart transplants. They both died, but one after about 5 months and one after about 4 months. And we're still trying to make them survive longer. These are organs from pigs that have been genetically modified to try to reduce the human body's immune response to the pig organ, and also do things like make sure the pig heart doesn't grow too big. This is not yet clinically available. This is still in experiments, but they're experiments that are making a lot of progress. We also are trying to figure out new ways to make new human organs. Some of those are using pigs or sheep. So there's a scientist named Ispuza, originally from Spain, who is trying to make pigs that have human kidneys. The way he does that is take very young pig embryos, change their genes so that those embryos will make pigs that do not have kidneys, but add to those embryos human cells that will make human kidneys inside the pig. This has not yet been done in humans. It has been done in rats and mice. There's no obvious reason why it couldn't ultimately be done in humans, but biology is uncertain. And we do know that humans are not mice and are not rats, but that would be another way to deal with the organ shortage. And then still another way to deal with the organ shortage is to just grow human organs. We have learned, thanks to a Japanese scientist named Shinya Yamanaka in 2007, how to take skin cells and give them a set of chemicals that turns them into things that are like human embryonic stem cells, cells that are pluripotent, that can turn into any other cell type. Now, scientists disagree how many cell types humans have. Some scientists think we have 300 cell types: skin cells, brain cells, hair cells, liver cells. Some scientists think we have 300,000 different cell types. But one thing we know is human embryonic stem cells can make every human cell type because they made us. We have every human cell type, and we're the product of effectively human embryonic stem cells. These induced pluripotent stem cells seem to do the same thing, with the added advantage. Let's say Luis has had a heart attack. Bad news. The good news is he's survived, but the bad news is his heart is weaker. He can no longer run the marathon in under 3 hours the way he used to be able to. He needs a heart transplant. If we made a new heart for him out of embryonic, an immune system would attack that heart. If we took skin cells from his wrist, from his hand, turn those into induced pluripotent stem cells, turn that into a new heart, and we put that in Luis, his immune system would say, welcome home. It's his same DNA. The immune system would recognize it and would think it was his and would not attack him. So I've given you 6 or 7 different ways we might be able to deal with organ shortages. Some of them are near term, things like keeping organs that have been removed viable longer. Some of them are long term. I don't think we'll be able to make Luis a new heart for a good 30 years. So take care of your heart for now. But in 30 years, if you need a new heart, we might be able to build you a new one from your own skin cells. What's amazing about this is those are a bunch of different possibilities, not one of which was available or even very plausible 25 years ago. I'm confident that one or more of them is going to work. If I knew which one was going to work, I would become rich. I don't know which one is going to work, but some of them are going to work. And that's just one example of the ways that the new technologies are giving us more plausible options to relieve human suffering. Let me talk a little bit about genetic diseases. So consider cystic fibrosis. So cystic fibrosis affects about one person in 20,000. 40 years ago, people with cystic fibrosis would have an average life expectancy of about 9 years. It is mainly a disease that kills people through their lungs, although it also affects other parts of the body. Every decade, the life expectancy got longer because we got better at treating it. And so now people with cystic fibrosis have a life expectancy of about 40, but it's not an easy life. They have lots of infections. They spend a lot of time in the hospital. They have to get treatment very, very frequently. Well, there are ways we could fix that, some of which are on the edge of being approved. It's a genetic disease. It's caused by a mistake, a misspelling in one gene, the CFTR1 gene. You could try to take a person with cystic fibrosis and use gene therapy to edit inside their lungs the gene that's broken to make it work right. I have a colleague here at Stanford who's working on just that fix. You could also try to do the same thing with editing embryos. If two people both have cystic fibrosis, they want to have a child that doesn't have cystic fibrosis. Any embryo they make would be a child that would have cystic fibrosis because they both have two bad copies of the gene. The child has to get one copy from each of them. It'll get two bad copies. But then while it's an embryo, before it has been put inside a person's uterus, you could edit that embryo to fix the gene. There are other things. One of the treatments that's already been approved for sickle cell disease, a nasty blood disease. It's caused by a mutation, by a bad version of the gene that makes globin, which is the part of which is a big part of hemoglobin. Hemoglobin is the molecule that carries oxygen in your blood, takes carbon dioxide out. People with sickle cell disease have misshaped bad hemoglobin because of a change in the globin gene. One approach has been to fix the globin gene through standard gene therapy. But it turns out that before we were born and when we were born, we have a different globin gene, fetal hemoglobin, that only works while we're a fetus or when we're an infant, and then it turns off. Well, one gene therapy that's already been approved doesn't fix the adult hemoglobin gene. It turns back on the fetal hemoglobin gene, which works fine in adults. Then there are ways of using what's called RNA interference to turn genes on and off. You don't change the spelling of the gene, you change when it's turned on and off to fix disease. But there are also the traditional things like small molecule drugs. And in I think the greatest medical miracle, except maybe for the COVID vaccine, of the last 5 years, people with cystic fibrosis, 90% of people with cystic fibrosis. And in a country like Argentina, I would guess that you probably have about 5,000 people, 10,000 people with cystic fibrosis in Argentina. People with cystic fibrosis, 90% of them can live without any symptoms by taking a pill that has 3 drugs in it. We understood how to make that pill because we learned more about how the disease works, thanks to advances in stem cells and advances in genetics and so on. But without having a very high-tech kind of treatment, 90% of people now live normal lives who have cystic fibrosis. It's miraculous. I'll give you one more example. Reproduction. A lot going on with reproduction. So People have always, it's funny. There are 8 billion people in this world. When my son was born 36 years ago, there were 5 billion people in this world. When I was born 72 years ago, there were just over 2 billion people in this world. 2,000 years ago, there were 100 million people in this world. Sounds like we reproduce pretty quickly, but in fact, among mammals, humans are actually pretty bad at reproducing. It takes us a long time and we're not very effective. Dogs and cats, cattle and horses, they get pregnant easily and they give birth easily, but infertility is a big problem among humans. And you can go back all the way to the Bible and Abraham and Sarah. Infertility was a problem for Abraham and Sarah. So infertility has been around for a long time. Starting about 60 years ago, some forms of infertility could get treated by artificial insemination, by taking sperm from a man and putting it into a woman with a syringe. But that wasn't very many cases of infertility. In 1978, Louise Brown was born, the first child ever born from in vitro fertilization. And suddenly, lots of people who couldn't get pregnant, who couldn't have children because the egg and the sperm weren't meeting or weren't meeting in the right place, suddenly could have children. Not easily, not cheaply, but they could have children. And today, in most countries, I don't know what the statistics are in Argentina, but in the United States and in most of Western Europe, 2 to 3% of babies born every year are born as a result of IVF. About 10 million people are alive today who would not have existed except for IVF. Well, that's a big deal, especially if you want to have children. But it doesn't help everybody. So, for example, what happens if you had you're male and before you went through puberty, you had a disease with a high fever? There's a very good chance that you would be sterile, that the disease killed off your sperm cells or the cells that were going to become sperm cells. Mumps is a classic disease for this. IVF can't help you have children that are genetically yours if you don't have sperm. Or you're a woman who had ovarian cancer and your ovaries were taken out. IVF can't help you if you don't have ovaries and you don't have eggs. Or you're a woman who's 45 years old and you had eggs, but your eggs no longer work. Well, there's a new process that has not yet worked in humans, has worked in mice, called in vitro gametogenesis. So making gametes, eggs or sperm, in glass, actually in plastic, but we call it in vitro. Again, you go back to the skin cells, you take the skin cells, you turn them into induced pluripotent cells, but rather than using those cells to make a new heart for Louise, you take those cells to make new sperm or new eggs. And so people who can't have children because one of them doesn't have eggs or sperm, can't have genetic children. I mean, I think for many people, adopting is wonderful, or using a sperm donor or an egg donor is wonderful. But there's still many people who really want to have a child that's genetically half theirs. So in vitro gametogenesis gives them the opportunity to do that, which they wouldn't otherwise have. But it creates some other opportunities. You could potentially have a woman who is 80 years old become a genetic mother, or a boy who is 8 years old become a genetic father. You don't have to wait for puberty. You take skin cells, you turn them into sperm. You could make eggs and sperm from somebody who has been dead, but whose cells were carefully frozen. You could do all sorts of things. The Japanese, who are leading in this research with mice, have made eggs from a male mouse. And they've combined eggs from a male mouse with sperm from a male mouse to make healthy mouse pups with 2 fathers and no mother. Who would want to do that? Well, there are a lot of gay and lesbian couples who would find that very exciting. So this is something that would relieve some human suffering. I think people who desperately want to have children who can't are suffering. This is a relief to them, but it raises new questions. Among other things, let's say, rather than a cup, let's say this is a can. I have a drink. I finish the can. I throw it away. On the lip of the can, there's some cells of mine. What if somebody picked up that can out of the trash, got my cells off it, turned those cells into induced pluripotent stem cells, turned those into sperm, and made me a father without me ever knowing it. Now, I don't think anybody would want me as a father without ever knowing it, but if you were Lionel Messi, you'd have to be very careful about where you threw out your beer cans or your Coke cans, because I think lots of people would like to have sperm from Lionel Messi. So wherever there are new opportunities, there are new downsides. And then two other possibilities, well, three, with reproduction, and then I'll move on to something else. One is selecting embryos. This has been possible for 34 years. The first child who was born as a result of embryo selection is now 34 years old, and she's a scientist. You have to use IVF for this. So you make, say, 10 embryos through IVF. When they're 6 days old, before you move them into a woman, you take a few cells off them, a few out of their 200 cells, take 5 of their 200 cells, do a genetic test on those cells and say, aha, this embryo would have cystic fibrosis. This embryo would have sickle cell disease. This embryo would be a boy. This embryo would be a girl. And the parents then decide which embryos to move into a uterus to try to make a baby. That's been done. Currently, among people in the United States who are doing IVF, about 80% of them go through this process, which is called pre-implantation genetic testing. Now, it's either good news or bad news. It can't tell you very much. It can tell you whether or not the baby would have one of 6,000 genetic diseases. All 6,000 of those diseases are rare, but when you multiply 6,000 times rare, you get about 1 to 2% of all births. So you could know whether or not your embryo was going to be the 1 or 2% who had the bad luck to have this genetic disease. And you could tell boy or girl. And you could tell light hair or dark hair, light eyes or dark eyes, with maybe 80% accuracy. We can't say black eyes, dark brown eyes, light brown eyes. We can't say blue eyes, green eyes, gray eyes, but light or dark, we're pretty good on. We can do that already. And that's getting cheaper and easier. The rate limiting factor for that is IVF, which is not cheap, easy, or entirely pleasant. But if you can do IVG, taking skin cells and turning them into sperm, and more importantly, eggs, there goes 80% of the cost of IVF and 99% of the risk and discomfort of IVF. So it could be used much more. Parents who don't need it for fertility could use it to do embryo selection. This is either good news or bad news, depending on how you think about it. It's not going to lead to super babies because we don't know the genetic variations that cause super babies. There probably aren't any. Or to be more accurate, there are probably 1,000 that have to be combined in a particular order. But it could lead to somewhat healthier babies and babies that cosmetically look more the way the parents want. We could also take embryos now and edit them and change the genes in the embryos. Hu Zhonghui, a Chinese scientist, did this 6 years ago. He ended up in prison in China for 3 years. He did it in a grossly unethical way. No one else thinks that this is ready to be used, but there are people who think that if it can be proven safe and effective, it should be used. That's another way to make babies that isn't here yet, but could be here soon. Another one, and this is one that I think is relevant to Argentina. I believe your president has 5 cloned dogs, or 5 dogs cloned from one dog, Conan. Human cloning has not been done as far as we know. We don't know whether it would be safe, but we now know that it could be done. For 11 years, we've known how to clone human embryos. So maybe we could clone Luis. Now, would we get Luis? No, we would get a baby. We would get a baby who would look a lot like the way Luis looked when he was a baby. But he would not yet be a professor. He would not be hosting talks. He would be a baby. Would anybody want to do that? Well, it's a big world with 8 billion people in it, and somebody probably would want to do it. And I'm a little surprised nobody has tried. And then the last of these reproductive changes, ectogenesis. Making a baby from start to finish outside a human. This has been talked about for 100 years. We aren't close to it yet, but I recently realized that there is a way we could get close to it within, say, 10 years. A problem has been trying to grow a uterus, trying to grow something that will feed everything from the little embryo to the 9-month fetus, have a placenta, have all the support that that pregnancy needs. But we now have uterus transplants. Over 100 uterus transplants have been successfully performed. 70 of them have led to live children. The uterus turns out to be a pretty resilient organ. So why don't we do this? Take a donated uterus from a cadaver, a corpse, or from a woman who's getting a hysterectomy, take it out of the body, hook it up to a machine that keeps it alive, that pumps blood and oxygen and sugar through that uterus, and then put an embryo in it and see if the embryo will grow for 9 months. Now, right now, that would be grossly unethical because the risks are unknown, but start by trying it with monkeys or pigs or sheep. And if it works really well with monkeys, then within 10 years, somebody will try it with humans. So again, lots is happening. Some of it is here, some of it is almost here, some of it is a ways off, but might be here. So these are changes that are happening. Opportunities are coming up. Let me go to the bad. Because what I've been talking about, to me at least, is the good potential ways to relieve human suffering and also to learn more. The bad depends on how it's used. Every technology can be used for good purposes or bad purposes. When I went to Argentina, I did not flap my wings. I went in an airplane. Jet airplanes are good for going long distances. It turned out we discovered in America on 9/11 that they were also good for bringing down skyscrapers if you ran them into skyscrapers. Jet airplanes are good, but can be used for bad. In my own personal ethics, what I worry about most in terms of the bad uses of biosciences are three things. Safety, used in a way that's unsafe, so danger and risk, harm, used in a way that is exploitative, and used in a way that restricts people's liberty. Other people worry about other things. Other people may worry that the technology is unnatural or is dehumanizing. I understand those concerns. I don't share them, but I don't think they're unreasonable concerns. They just come from a different ethical background than mine. For me, the biggest worry is, sure, you use this new system to make babies without people having to be pregnant, but the babies turn out to be disabled often. Or there's some long-term change in family structure because if a woman hasn't had to be pregnant for 9 months with the baby, she doesn't feel the same sort of attachment to it. That might happen. That would be a bad thing. Or yes, you make organs from pigs and use them in humans, but after 10 years, the human dies terribly. That would be a bad thing. Another risk is not just accidents, things, unexpected negative consequences, but intentional negative consequences. Genetic engineering makes many good things possible, but it also makes it easier to do biological warfare. Neuroscience interventions make many good things possible, changing people's brains in ways that make them healthier, but it also potentially makes it possible to change people's brains in ways that make them less healthy. Let's say we figure out how to affect pain in the brain. When I do this, it feels like it hurts here, but it really hurts here. The pain is in the brain. If we learn enough about where the pain is, and we are learning that, we could shut the pain off. But if you're a bad guy, maybe you want to induce pain in people as a way of torture. Things that can be used for good purposes can often be used for bad purposes. Harm, direct harm, is a bad potential use. Another bad potential use is exploitation. It's using them in ways that exploit people. My least favorite example of this is in the United States, there are 500 clinics that sell stem cell treatments, none of which have been approved, none of which have been proven safe, all of which are very expensive. There are 500 clinic directors who are getting rich, selling people things that probably don't work and might hurt them. That's bad. Exploitation is a bad use. Then there are issues around coercion. So, say, forcing all parents to do prenatal testing of their fetus, or forcing parents to give birth in a certain way, or forcing people to go through mind personality changing neuroscience techniques that make them less religious or more liberal or more conservative, changing people without their consent. Sometimes that's a good thing when you're trying to help somebody who's mentally ill, but sometimes it's a coercive bad thing. These offer risks to liberty. What do you do with a technology that can have wonderful consequences and terrible consequences? You try to regulate it. Will you regulate it perfectly? No. One way to regulate it is to ban it. But for a lot of these technologies, banning, you give up so many potential good things that it's probably not worth it. But it depends on how well you can avoid the bad things. And trying to draw that line and have an effective, enforceable way to get the benefits from the biotechnologies while minimizing the harms. That, I think, is the task for the next generation of people working in law and biosciences or ethics in biosciences, the next generation of legislators, and presidents and prime ministers and regulators. And it's not going to be an easy one. And then finally, the incredible. So I say this with a little bit of caution. There's a famous quote, often attributed to the Danish physicist Niels Bohr, though probably it's a Danish saying from someone else. It's always hard to predict things, especially the future. It's a good line. I should have memorized it in Spanish. Siempre es difícil adivinar las cosas, especialmente el futuro, something like that. I hope that was close enough. It is, of course, wrong. It's easy to predict the future. It's just hard to be right when you're predicting the future. I'm going to predict the future in two ways. I think I'm right, but I might not be. One is people are excited by and worried about brain-to-computer interfaces. Elon Musk wants everybody to be able to connect directly to the internet in their brain and be able to telepathically talk to other people, brain to brain. I don't think that's ever going to happen. Brains are too complicated. There are others who think Ray Kurzweil and the singularity is near, thinks we will be able to become immortal by uploading our brains into computers. I don't think that's ever going to happen. Brains are just too complicated. Forever is a long time. I may be wrong, but I would not spend a dime on trying to do that goal. Brain to computer interfaces to help people who are quadriplegic, who are paralyzed, yes, that's a good goal. But to try to let people get the internet in their brains when I don't think there's any way it could happen, and I'm happy getting the internet on my laptop, I think that's impossible, and we shouldn't waste time and money on it. Another one is immortality. So there are a lot of I live in Silicon Valley. Silicon Valley has a lot of billionaires. Some of those billionaires think they should live forever, and they've got lots of money to spend to try to live forever. I don't think they're going to succeed. The body has so many things that can go wrong. If you keep the heart alive, well, then there are the lungs. And if you keep the lungs alive, then there's the liver. And if you keep the liver alive, then there's the immune system. So far, the oldest person known only made it to 122, and she's the only person ever to get past 119. Almost nobody lives past 110. Maybe we'll get to 125, but we're not going to live anywhere close to forever. The US Patent Office has a rule. They refuse to even review a patent application involving perpetual motion machines. I have sort of an informal rule. I don't even want to think about anybody saying they're working on immortality because it's something everybody wants, has lusted for. Supposedly, Ponce de Leon discovered Florida when he was looking for the fountain of youth. It is literally incredible. Incroyable. Not going to happen. So that's the good, the bad, the incredible. In conclusion, every audience's favorite two words from every speaker are in conclusion. In conclusion, this is a wonderful time to be alive. It's also a terrible time to be alive, but it's a wonderful time to be alive, especially if you're interested in bioscience, because the science is both amazing and hugely important. Yes, computer science and AI, that's really wonderful and amazing too. But we are biological organisms. When we learn about biology, we learn about us. And that is more directly important, I think, to us. Not that we could do the biology without the computers, but the biology is amazing. And as a lawyer, as a law professor, it's law and ethics and culture and politics that are even more important than the science. The science tells us what we can do, what we might be able to do. But the science can't tell us what we should do, or how we should do it, or how we should regulate it, or what we should allow and what we shouldn't. And that will differ from person to person and culture to culture. Argentina will allow some things that maybe Brazil wouldn't, or the United States will allow some things that the Vatican City certainly wouldn't. So there's going to be variation, but it's going to be those of us who work on these issues on the social side of the issues, but who know enough about the scientific side, who are wonderfully placed to try to help make the world change in good ways, but not in bad ways. And that's where authors, scholars, teachers, speakers, journal editors, those of us who work in law and the bioscience or bioethics, that's our role. It's an important role. I think it's a noble role. But I would also say one of the things that I like best about it is it's a lot of fun. Life is short, fun is good. If you can do work that you think is important and that you enjoy, that's what you should aim for. That's what you should aspire for. And I recommend working on the ethical, legal, and social issues of the biosciences is a great way to do that. I think I've
Thank you very much, Professor Hank Greely.
Muchas gracias por su atención y buenas noches.
Bueno, muy buenas noches. Gracias. Thank you very much.
Good night.
Thank